Welcome, everybody. Let us start this seminar. So, at Molecular Biology of the Cell, we have a Quantitative Biology Special Issue every year. Call for papers normally comes out in the fall. So this year, issue just came out with almost 20 exciting papers that cover many topics from cytoskeletal dynamics to cancer cells, to morphogenesis, to image analysis and microscopy tools. We at Molecular Biology of the Cell understand quantitative biology very broadly, as not really different from traditional cell biology. We think that quantitative cell biology embraces any use of numbers, quantitative experimental data and models to achieve predictive understanding of the cell. As opposed to just qualitative experiments. Not that there's anything wrong with qualitative experiments. As we all know, there were pioneering discoveries made with those. So without further ado, let me start with introducing the first speaker. We have three exciting talks today based on three papers published in the current issue. So the first speaker will be Juan Saicedo, who is a Schmidt Fellow at the Broad Institute at MIT at Harvard. Juan holds PhD from the National University of Columbia, and he worked at Google Research, Microsoft Research, Queen Mary University of London, University of Illinois at Urbana-Champaign. So he will talk about cell painting. Which is a very non-trivial non-gene profiling assay that captures features of the cell based on microscopy and uses deep learning to predict lung cancer variants. So Juan, please go on. Just a brief note that put all your questions in Q&A at the bottom of the screen. And we'll read them out after each presentation. So a reminder that the talk should be hold to about 15 minutes and 5 minutes for questioning. Juan, welcome.
Great. Thank you so much, Alex, for the kind introduction and the invitation to participate in this webinar. It's my great pleasure to be here and share with you our research using cell painting for phenotyping variants of unknown significance. Can you see my screen here?
Yes.
Okay, great. So I am talking on behalf of a large team at the Broad Institute who was working together in order to understand how we can classify the impact of variants in cancer, and you know, all the work that I'm presenting here is a collective and joint work with multiple people. So just to give an introduction, the number of variants of unknown significance is growing every year. And this is true not just for cancer, but for many other diseases. And what you can see here in the screen is the registered genetic tests in the x-axis. And in the y-axis, you see the total number of variants that are reported after testing tumors or patients for specific diseases, including cancer, heart disease, or other type of diseases. And the size of the points indicates how many of those variants that are reported are of unknown significance, which means those that we are not particularly certain about the function of those variants or the impact of those variants in the development of cancer specifically, which is our interest in this project. And what this means is that we are observing a bunch of mutations in the sequencing of tumors, and we are not sure if those should be targeted for designing treatments, or maybe they are just side effects of the cancer. and we can ignore them in order to focus our resources and strategies in something that is going to be more fruitful for patients. So our research question here is, can we identify the functional impact of those multiple variants that are reported in the clinic or by the clinic and in the literature, and try to identify whether they are impactful or they are neutral mutations in order to classify them and inform the design of treatments. And for that, we used the cell painting assay, which is an unbiased morphological profiling assay that allows us to capture cellular morphology in a comprehensive way. The assay has 5 channels, which are the result of staining the cells with 6 different dyes that target basically most of the cellular compartments in order to get the structure of the cell and quantify their morphology. And this assay was used to scan a large number of mutations in this project. Usually, when researchers are investigating the impact of variants, they follow a more functional and targeted approach, which is definitely kind of the ground truth or gold standard in order to determine whether the impact of variants is neutral or not. But in our case, what we wanted to do is to accelerate the pace at which we classify the multiple number of variants that are reported every year in multiple diseases. So in this experiment, we evaluated or analyzed a total of 350 mutations. And what you can see here in the screen is example single cells extracted from larger populations of cells in the images, representing some of the wild type genes in our study. And we have a total of about 5 million single cells in this study, and we used overexpression in order to introduce the variants of interest in multiple replicates. So this is kind of the experimental setup. And of course, our next step is going to be to transform those images into quantitative data that we can use for analysis. So this is kind of the workflow that we follow in order to transform images into data. So we start with the perturbation, which, as I mentioned, is an overexpression perturbation in which we introduce the mutations to the A549 lung cancer cells. And then we proceed with the staining in order to capture images using the cell painting protocol. And we get the images and we proceed to do image analysis. The image analysis in this case involves cell segmentation and feature extraction from each of the cells that we identify in the cells. After that, we aggregate information from the population of cells and we create profiles or signatures that identify what is the average response of the cells after doing the perturbation. With those profiles, we proceed to do data analysis. And here are some example images with one reference gene and one mutation that we are looking at. And our goal is to automatically and quantitatively identify any differences in the phenotype revealed by the cell painting assay. We found that approximately 83% of the variants and wild type genes that we had in our study can be clearly discriminated or differentiated from random perturbations in the set of experiments that we did, which indicates that the data that we collected is of high quality. It basically indicates that the effect of the overexpression experiments can be replicated across multiple replicates and it's consistent. So what we observed then is what is the structure of the relationships between the multiple genes and variants that we are analyzing here. And this is the matrix of correlations between the profiles of each of the variants and genes in this study and how they correlate to each other. So we were pleased to see that the morphological feature space is able to cluster the variants together when they belong to the same reference gene. So for instance, this branch over here corresponds to most of the variants from the BRAF gene. And we see a lot of structure in this dendrogram, indicating that cell painting and morphological profiles specifically can differentiate the minor or subtle differences in phenotypes in order to cluster these in a meaningful way. Looking at another representation of the same data using UMAP plots in which we project the same basically data representation in a lower dimensional space, we again see that most of the variants, for instance, in this case of KRAS, are related to each other. This basically indicates that the differences or the morphological changes that happen from the wild type gene, which is in green, with respect to the mutants, which are colored in orange, are noticeable by the assay. So we can notice the differences, but they are also not major changes in morphology. Like those differences can be quantified, but they are not, making one of the KRAS mutations look more similar to EGFR, for instance. They don't travel that much in the feature space. They remain closely together, and we want to quantify whether those changes or those variations that we observe around the reference genes are meaningful in terms of the impact of cancer mutations. So in order to determine whether a variant is impactful or not, we used an algorithm which was previously developed for gene expression analysis of this same, type of problem. So basically what we do is we interpret the correlations between replicates of the same perturbation as probability distributions. So here is kind of the decision tree, and we have the reference gene in green, and we compare the distribution of the correlations against the correlations of the variant in orange. And we also look at the cross correlations between reference gene and variant in order to see whether they have something in common. If those distributions are all of this, all of them the same, we can say with a certain degree of confidence that they are, that the variant is a neutral mutation because it doesn't have any difference with respect to, the reference gene. And this is kind of an example of a neutral mutation. Most of the correlations across the reference and the variant don't have any meaningful difference. But if you look at an example of gain of function mutation in which the reference gene has certain correlations and the variant has a different correlation structure, we can infer that this variant is probably a gain of function mutation and it's not neutral, or it's doing something different than the original reference gene. Same with the loss of function mutation and change of function mutations. We can interpret those correlations and make an inference about the status of those variants. And here are the results for a few more variants in which we see, for instance, that all of the variants in the BRAF, well, some of the variants in the BRAF family are behaving as gain of function mutations. And in order to give you a little bit more context to interpret these plots, we have a couple of p-values in the decision tree. One that tells us the likelihood of that variant to be impactful with respect to the reference gene, and that value is in the x-axis. And in the y-axis, we have another p-value that tells us whether this is a gain of function mutation or a loss of function mutation. And this is in the y-axis what we have. If you see any blue or p-values down here, it means that it's likely to be a loss of function mutation. But if you see the points up here, it means that it's probably a gain of function mutation. If the points are close to the origin of the plot, it means that they are neutral type of mutations. And we can see, many mutations doing many different things as well, indicating that the quite diverse phenotypes that are induced by those mutations may have different types of impacts. Another interesting thing is that we can perform single cell analysis using cell painting because the images have the information of every single cell, and we can quantify that as well. This is an example of a BRAF mutation, well, actually two BRAF mutations, B600E and W450L. Both of them were classified as gain of function mutations according to the decision tree that I described before. But when you look at the single cell heterogeneity that those mutations are inducing, we can see that they move to different regions of the morphological feature space with respect to the variant. So in this plot, the green cells are the reference gene, the variant cells are in orange. And as you can see, there is a difference between the populations of cells, but they move into different regions for the two variants that we're looking at here. And what that may mean is that they are disrupting the underlying function of the gene in different ways. And this is also something to investigate even further as well. We validated the predictions that we can make using cell painting with transcriptional profiling. There was a subset of those variants that we used in our analysis that were previously characterized using gene expression profiling. And we see a high correlation between the signal that we obtained with cell painting and transcriptional profiling. So in this specific plot, you can see that in the x-axis, we have the signal strength of L1000, which is a transcriptional profiling platform for some of the variants that are depicted here. And each point is one of the variants that we have in this common subset. In the y-axis, we have the signal strength of cell painting. And what we can see is that anytime that we see one variant that has high signal in the transcriptional profiling platform, we also are likely to see a high signal in cell painting. They are very correlated to each other. And doing a canonical correlation analysis between the two types of data also reveals quite an agreement between both types of platforms, which indicates that gene expression and morphology are highly correlated. They are not exactly the same. They are not revealing exactly the same information, but it makes a lot of sense that the variations and differences that we see in transcriptional profiling are also expressed in morphology, and we can capture them with imaging. We believe that they are kind of complementary as well, and this is also something that we are interested in investigating further in the future. If we compare the prediction...
Time is pretty much up.
Oh, okay, sorry. I'm going to wrap up. So cell morphology is useful for variant impact phenotyping. Image-based profiling allows us to explore single cell heterogeneity, and cell painting predictions are very well correlated with transcriptional profiling predictions. So thank you so much for your attention, and I'm happy to take any questions.
Excellent. So we have 3 questions. Actually, I think maybe 4 questions. Yeah. Please, Juan, read them in Q&A and answer, and we have only like 3 minutes.
Okay, great. So question 1, single mutation may not have a phenotype, whereas it may impact in combination with others. That is a good point. We were looking at single mutations only, and this subset of mutations was selected from a list of high frequent mutations observed in the clinical reports. But it is true that combinations of mutations may have different types of impacts, and we were looking only at one. It says, what does BRAF-REF refer to in the slide? In the slides and also in the paper, we name the wild type genes as reference genes. And that was some sort of agreement that we made while writing the paper, but you can interpret this as wild types, basically. Do you normalize the phenotypic outcome with expression level of reference versus mutants? No, we actually, in the morphological profiling pipeline, we normalize with respect to control cells. And control cells in this case are cells that didn't have any overexpression perturbation. So they are called empty cells because they are kind of the natural looking A549 type of cells. And the final question, oh, there are a couple more. Are there names given to the morphology of the cells for each group of phenotype, for example, round or protrusive? This is a great question. We used a deep learning-based feature extractor, which does not have names for the individual features that we have. And we are not categorizing those with explicit names either. So I think there is still a lot of work to be done in the image-based profiling field in order to interpret those differences that we observe, where exactly can be those detected and what they mean specifically. Something that we are very sure and satisfied about is that the technical variation was removed successfully as part of the normalization process as well. Okay, I think with that, I also answered the final question, which is what are the features that we are capturing? And as I mentioned, they are deep learning-based features. So I think that's all.
Thanks very much again, Juan. And
Thank you so much.
Now Diane will take over for the second and third talk.
Great.
Yes, thank you, Juan. That was great. Our next presenter is Charlotte Kaplan. Charlotte received her PhD at the ETH in Zurich, where she performed some really beautiful work using super resolution microscopy to study how septin filaments assemble in cells. But then moving on to her postdoc work in the Druben lab at UC Berkeley, she's now applying high resolution imaging to the study of actin network organization during clathrin-mediated endocytosis. And so she's going to present her work from her postdoc today in the talk entitled Load Adaptation by Endocytic Actin Networks. Charlotte?
Yeah, thanks very much, Diane, for the great introduction. And also, I want to thank MBoC for this really great opportunity to present my work here from my postdoc. So first of all, now, okay, I need to use the mouse. First of all, I want to basically walk you through the process of clathrin-mediated endocytosis in mammalian cells. So this is a process that transports cargo across the plasma membrane of cells. And what you see here depicted in this cartoon is the plasma membrane outlined here in white. And then cargo binds from the extracellular space to the plasma membrane. And this recruits a machinery, a protein machinery, including the clathrin protein. The clathrin protein then forms a lattice at this initiation site, and this, together with the protein machinery, brings the membrane into an invagination that later on in the process forms this vesicle structure, and the plasma membrane then is separated from this vesicle structure, and this vesicle is transported further into the cell to its target organelles. So one really prominent protein here, which is supporting this process, is the actin cytoskeleton. And the actin forms this network around the clathrin-coated site and basically provides the force for the plasma membrane invagination process against the underlying plasma membrane tension. So this relationship was established between the relationship between the actin network function, providing basically force against the underlying plasma membrane tension by Steve Boulogne. He pioneered in this work from the Kirkhausen lab. And here in this experiment, he used the adapter protein 2 labeled with fluorescent dyes and fluorescent microscopy, and measured the lifetimes as a readout of the process of clathrin-mediated endocytosis in MBoC cells. And he, in this experiment, inhibited actin dynamics with a drug. This is jasperkinolide, and at the same time, he increased the plasma membrane tension of cells. And he performed this by using hypotonic shock treatment on these cells. So basically, when you put cells from an isotonic media into a hypotonic media with a lower osmolarity, you have a water influx, and then the cell membrane swells, and this increases for a short time the plasma membrane tension of the cell. So under normal isotonic conditions, you see here in this plot that counts the arrested pits of clathrin-mediated endocytosis over the time of the movie, that jasperkinolide treatment increases the number of arrested pits to 8% over the time of the movie acquired. When he then increases the plasma membrane tension by hypotonic shock treatment, he really gets a 10-time increase of stalled pits under really high hypotonic treatment. So this really shows that actin is really required for the force production here against the underlying plasma membrane tension to form this plasma membrane vesicle. However, from these live cell experiments that are really beautiful, we don't really get the information about how the actin network on the ultrastructural level achieves this force production and provides the robustness for this process of clathrin-mediated endocytosis. So that's why I asked the question in my postdoc work: how does the ultrastructural organization of the actin network adapt at endocytic sites under elevated plasma membrane tension? And for this, to reveal the organization of the actin cytoskeleton that is associated with these clathrin-coded structures in mammalian cells, we use three-dimensional dual-color super-resolution imaging. And this was a great collaboration with the lab of Chris Xu at UC Berkeley. And here in this region of interest from a cell, we use skin melanoma cells here in this work. You see a super resolved actin cytoskeleton and the clathrin-coated pits. The actin cytoskeleton here is labeled with the fluorophore of Alexa 647 of this wavelength, and then we labeled the clathrin-coated structures with CF with cyan dye of 680 nanometer wavelength. Absorption spectra. So here you see these actin networks, and they are also interacting with these red spots, which highlight the clathrin structures. And I just want to show you a zoom into these structures, because this is really where what interested us in our study. So here you can see this elliptical structure of a clathrin-coated pit, and then we really wanted to quantify the actin network that is basically associated or even covering in this XY projection the clathrin-coated pit. So for quantifying only really this small part of around the actin network and around the clathrin-coated pit, we cropped a really narrow square around these structures, and then we basically generated side projections of these Kaplan-coated pits to then be able to quantify the height of the actin network and its organization. So we selected from these images over 700 of these Kaplan-coated structures. And here I want to show you 3 examples of actin networks that are associated with these. So just for your orientation, you have here in the upper part of the image, the Kaplan structures that points into the direction of the cytoplasm. And then in the lower part, this is the extracellular space. So in my first example, I show you here a really narrow actin network at the base of the Kaplan-coated pit. In the second example, the actin network covers the Kaplan-coated pit already up to 50%. And then we also find many examples where we have a full coverage of the Kaplan-coated pit by actin here. So from this quantification that we could do, we could derive that the actin network in this process of Kaplan-mediated endocytosis in mammalian cells grows from the base of the Kaplan-coated pit to the tip. So now with this established pipeline of super resolution imaging of these 2 structures, with selecting hundreds of structures and then quantifying the organization of the actin network, we then next performed this experiment in cells that were under higher membrane tension. To first make sure that these cells in our experimental settings indeed had increased plasma membrane tension, we collaborated here with the lab of Alba Des Munoz to perform membrane tether pulling experiments on the cells to measure the plasma membrane tension. You see here under isotonic conditions that we are getting forces around like 30 piconewtons, and then under hypotonic conditions we really can increase significantly here the plasma membrane tensions to over 45 piconewtons in mean. So this condition then we chose for performing under the hypotonic media treatment, the treatment of the cells for super resolution imaging. And then, of course, we again selected hundreds of Kaplan-coated structures that were associated with the actin and performed the quantification. And here in this plot, you see the actin height readout of hundreds of single Kaplan-coated sites. And on average, in median, we found under isotonic control conditions height of 130 nanometers that significantly increased when we increased plasma membrane tension in these cells on single Kaplan-coated sites to a median of 160 nanometers. And then then we also quantified the coverage of the actin over the code in percentage. And also here we found a 10% significant increase under increased plasma membrane tension conditions. So with these experiments, we could really show that there is an adaptive nature of the actin network to the increase of plasma membrane tension in individual endocytic events. So next we ask when the actin network is required under elevated membrane tension in these cells. So for this, we inhibited the actin network polymerization with the drug CK666. This is especially inhibiting the actin ARP2/3 network polymerization, and performed live cell imaging here on these SKML cells that express clathrin, the marker for endocytosis here, and dynamine. This is the GTPase that is involved in the late scission step of endocytosis. And under this live cell imaging, with his live cell imaging experiments, we found the conditions where we could inhibit actin polymerization, then also increase plasma membrane tension, and this significantly elongated the lifetimes of these clathrin-mediated endocytic events with an additive. This was an additive effect in comparison to the single treatments. And under this condition, we again performed super resolution imaging on these cells, especially, and this time only on the clathrin codes to then quantify their height, and this as a readout for the plasma membrane invagination. And here under only actin inhibition with CK666, we accumulated many more clathrin codes that showed a higher height. And this is consistent with earlier findings that when you inhibit clathrin, you stall the process of clathrin-mediated endocytosis in a later stage. But then when we increase the plasma membrane tension simultaneously by hypotonic treatment, we found many more codes that were smaller, which then means that the process is stalled earlier on in the process of plasma membrane invagination here. So with this, I want to summarize the findings that I could show you here in the seminar, but that we also published in our work. So here we propose a model in which we plot the membrane tension on the y-axis and the time of the endocytic events on the x-axis. And when we have really fast endocytic events, we propose that no actin network is required for providing more force to this process, because the assembly of the clathrin lattice itself provides enough energy, and also the proteins from the machinery that is helping the clathrin. This is basically enough for the membrane invagination progression, but also for the membrane fission step. If membrane tension increases to an intermediate level, then these the events elongate in time, and here actin becomes necessary in the later events of endocytosis in membrane fission, and as more assemblage in a kind of narrow regime around the base of the clathrin-coated pit. But then when the clathrin events really experience high membrane tension, they start really early on, because the energy of coat assembly and the proteins is not enough anymore to drive the membrane invagination further. Then actin assembles and then can cover the coat completely and provides more force due to polymerization to successfully provide a membrane scission and further vesicle transport. And I quickly want to highlight the 3 findings from our work. This is that I showed you with the super resolution images that the actin in mammalian clathrin-mediated endocytosis grows from the base of the clathrin-coated pit to its tip. Then the actin grows bigger at all geometric stages of clathrin-coated pit invagination. This is also was a very unprecedented finding, since it was thought that it's always really assembling at late stages and provides a burst and force there. And under higher membrane tension, actin is really required also in the early processes of clathrin-mediated endocytosis. And with this, I want to really thank the lab, my postdoc lab. David was a wonderful mentor during that time that I spent there, and also during the work there. Then Matt, and I want to thank Joe for being involved in this project, and also our collaborators, the XU lab and the Desmonos lab. Thanks very much.
Great, thank you, Charlotte. That was a beautiful talk. Just to remind people to please type your questions in the Q&A box, but I did see a question sneak into the chat, so I'll just read that out for you first. How is actin coverage measured, and how is this differentiated from actin network height? Are the two correlated with one another, or can they be distinguished as separate parameters?
So the actin network, we measured the height by projecting, I showed these side views, and then we basically projected these side views onto one axis. So we had and plotted, and basically you would then get a histogram of these of the pixel intensities, and then you can plot plot a line to this histogram, and then we had the readout of the 30th percentile of the thickness of this plot as actin height. Then to the second question, we actually, this is in the supplementary data. We also, in addition, measured the actin height of the cortex, of the neighboring cortex. And this, we didn't see any increase in the actin cortex height under increased plasma membrane tension, but it's specifically an increase of the actin that is associated with the clathrin-coated pits. Yeah.
Yeah, we still have a couple minutes, so if you want to look in the Q&A box.
Yes, increased. I just need to see where's the first question. Okay. Beautiful comment. Did you observe a difference in the shape size? Okay, sorry. Did you observe a difference in the shape size of clathrin-coated pits with increasing actin network? Actually, I didn't look at that particularly. I did measurements of the shape and of the size of the clathrin-coated pits, but only basically compared these from the isotonic conditions to the hypotonic conditions. But these results were not really clear. Yeah, but no, I didn't do this comparison. Then is the increased height with increased tension due to the time rather than tension? That is a result of tension-induced arrest or decreased rate in coated vesicle formation rather than due to tension itself. I mean, this kind of is like, I would say this is something, I mean, basically that's the reason. I mean, you due to tension, you elongate the process, and of course you give actin more time to polymerize when the polymerization is initiated. So this is something which just like is kind of a feedback loop in itself. So I would say yes, yeah. Then have you looked at this actin assembly in clathrin-independent endocytosis? Are there similar processes involved? I think this is super interesting. No, we didn't do that. Yeah. Then is the actin required to pull harder under increased tension? This is definitely something which one could answer with simulations. And I hope my former postdoc colleague Matt is more, kind of going into this direction to study this. So,
thanks for that. I think due to time, we're gonna have to move on to the third speaker, but you can type some answers in the boxes. And we'll check that out.
Thanks
All
very
right,
much.
thanks again. Congratulations on some really nice work.
Thanks.
Okay. So it's now my pleasure to introduce our final speaker of this webinar series, Jesse Aaron. So I've actually known Jesse for a while because, you know, science is a small world, and he did his postdoc at Sandia National Labs, where we overlapped in interest, where he was doing some really nice work developing hyperspectral and STED microscopy. After that, he moved on to Ventura, where he's an engineer for a while, developing 3D super resolution technology, before finally landing at Janelia Farms, where he is currently. And for our special issue, he and Chad Hobson, the co-author on this work, contributed a technical perspective, looking at combining imaging modalities and how that's important for cell biology research. So he will present his work entitled Combining Multiple Fluorescence Imaging Techniques in Biology When One Microscope is Not Enough. Thanks, Jesse.
Thank you very much, Diane. And thank you all for the invitation to give a quick talk here, and congrats to the other speakers. Yeah, I'm Jesse Aaron. I'm an application scientist at the Advanced Imaging Center at Genelia Research Campus. And I wanted to talk today a bit about this paper that my colleague Chad Hobson and I wrote. This was a technical perspective piece, so not a primary research article, but more of a piece with kind of a guide in mind to help people understand what are the benefits and challenges of combining multiple fluorescent microscopies together to try to get something that's more than the sum of its parts. And I want to start and talk a little bit about the motivation that we had for writing this paper. So a lot of times at the Advanced Imaging Center, we have people coming to us telling us, I really need to image my sample live for a really long time at high speed, multiple channels, maybe super resolution, maybe deep inside the tissue, and all of these things. And the problem with that is that, you know, this is exemplified by this figure here that we call our pyramid of frustration. And what we're trying to convey here is that if in any microscopy experiment, if you try to maximize some imaging parameter, say resolution or speed or what have you, it's always going to come at the cost of one or more of the other parameters. And so very often what we find is that no single microscope can really address a complex sample or a complex hypothesis. And so instead of throwing up our hands in defeat, what we can do is say, and we can draw from now more than ever, the vast toolbox that's available to us in terms of fluorescence imaging techniques, and actually combine multiple imaging techniques as a way to get around this pyramid of frustration. And for many of us that do microscopy sort of day in and day out, this might feel like a fairly obvious point, but as we thought about it a little bit more deeply, we realized, you know, there's a number of different ways that we can go about combining different microscopes, and that's what I'm showing here in this slide. And we came up with sort of 3 different classes, if you will, of microscope experiment designs. So I'll go through those. So the one on the left is certainly the most common thing that we see, and that's a scenario where you just have 2 samples that you want to image on 2 different microscopes, and you can image those in parallel. But it's not really until you've extracted some meaningful information or some insight from this data that you really combine the results of these experiments to tell a story that really no microscope by itself could tell. But very often people want to do something more complex. They really have the need to image their same specimen and really their same field of view within that specimen on 2 different imaging techniques. And so to do that, you know, that's what we call correlative light microscopy. But in order to do that, most of the time, if not all the time, you have to fix your sample at some point in this process in order for the data from one technique to be accurately spatially registered with the data from your other techniques. So while this is powerful, a drawback is that this usually prevents you from being able to image one of these techniques in a dynamic fashion. So that brings me to this 3rd experimental class that we talked about, and we call this dynamic multi-technique imaging. So like before, we have a specimen here that we image, we want to image using 2 different techniques, but we have it on a system that can actually dynamically switch between different imaging modes, or maybe even image 2 modes simultaneously. So what this kind of experiment can let you do is, yes, you can do this correlative imaging that I talked about before, but you can do it in time in both techniques. So I'm going to go through and give some what I hope are illustrative examples of each of these, and hopefully that can inspire people to extrapolate from and when they're designing their own microscopy experiments. So I'll start off with the simplest case where we have these multi-instrument but multi-sample kind of approaches, and a good example we thought was this paper that came out of Claire Waterman's lab, where they were looking at complement receptor mediated phagocytosis. And what they did was they imaged some of their macrophage cells engulfing these opsonized polymer microspheres using 3D spinning disk confocal, and it revealed this really interesting sort of reaching around mechanism where the particle, or the membrane reaches around the particle before it pulls it inside, which was interesting in and of itself, but it really spawned a lot of questions that they wanted to answer, including what was the nature of the actin cytoskeleton that was presumably driving this process to some extent. And they had sort of two questions that really competed against each other. One was, what was the nature of the actin flow as it's pushing the membrane around this particle, but also what is the nature of the actin structure itself? And if we think back to this pyramid of frustration, speed and resolution are inherently at odds with each other. So they did the smart thing and they split this experiment into two experiments. And so the first thing they did was they made use of fluorescent speckle microscopy, where they used a photoconvertible version of actin and photoconverted just a small portion of the actin, and where they can track these small puncta as the actin engulfs the particle here. And they found, interestingly, that the actin was pretty much unidirectional. It didn't, or at least there wasn't any retrograde actin flow in here. So this answered part of their question, but of course they had to sacrifice something in order to answer that question. And one of those things was that, well, they could only really do it in 2D in order to get enough temporal resolution, but maybe more fundamentally, there's really very little structure to be seen in these kinds of images. And so to get at these structural kinds of questions, they turn to structural illumination microscopy. So in here, with SIM, you can get much better resolution than you could, say, in spinning disk, but to do it in turf sim, in order to capture this process in time, they had to reorganize the geometry of their samples so that now they're doing a frustrated phagocytosis assay where the cell is settling down on a functionalized surface. But what it allowed them to do was to see very nicely the structure of the actin during this process. And so they could see things like really highly branched networks, indicative of an ARP2/3 mediated process, and interestingly, no stress fibers. So the role of contractility here seemed to be minimal. So I think this hopefully serves as a good example that by choosing the right imaging technique to match a specific question, they can build up a story, again, that no single microscope could tell on its own. But of course, the downside here is that these data sets were from different samples and even really different sample geometries altogether. And so that brings me to our second example here, where they make use of correlative light microscopy. And this was a study in PNAS where they were interested in looking at lysosomal transport along microtubules. And so what they did here was they labeled their microtubules and imaged them in a wide field microscope at relatively high speed. And from that, they were able to get the trajectories of their lysosomes. But what they wanted to do is see how is that related to the microtubule network in the cell. And so what they did was they very carefully fixed their sample on the microscope stage and actually did immunofluorescence staining of the microtubules. And then they performed single molecule localization microscopy so that they could generate this very high resolution image where they can resolve individual microtubules in the same field of view. And then what they could do then was map these trajectories onto specific microtubules in the cell. And so armed with this correlated particle tracking and super resolution data, they could start now making some inferences about how in some cases lysosomes can sort of pass under microtubules that are crossing their path if there's sufficient space, or maybe not if there isn't. And in some cases they could see lysosomes hopping from one microtubule to another. But they were careful to point out here that this fixation step that they did in the middle of their experiment could provide a source of error, right? Because maybe the microtubule moves somewhere between the particle tracking and the storm imaging, or maybe the fixation didn't perfectly preserve the structure in every place in the sample. So that brings me to this third case here, where we really want to do dynamic multi-technique imaging, where we're either dynamically switching back and forth between two techniques within the same system, or maybe even doing them at the same time. And so this is an example from a group in Hamburg, I believe, where they wanted to show how spinning disk microscopy can be combined with turf microscopy to both track a kinesin positive vesicle in 3D, but also to identify interactions that it was having with the plasma membrane. So that's what I'm showing here. So The green signal is their spinning disk signal, and it shows this vesicle moving in 3D within the cytoplasm. And then the turf data here is shown in magenta. And so what they were able to do is combine these two data sets. They were actually imaging these things both at the same time. And with the spinning disk, you can see they were able to track this vesicle as it's moving around, but only when the vesicle touched the plasma membrane did you actually see signal in the turf modality. So in this way, they could do two things at once. Essentially, they can track this vesicle wherever it happened to be, but they are also able to pinpoint when it was able to touch the plasma membrane. So again, a very elegant way to combine two modalities to get some information that you could not get with either modality by itself. So I hope these examples, as quick as they were, give people some inspiration on how they can extrapolate from here when they're designing microscopy experiments. But I want to try to generalize here a little bit and talk about microscopy experimental design. I think most of us are relatively familiar with what we call the forward flow in experimental execution here. This was a paper that we published a couple of years ago, but essentially I think it's fairly self-evident that you can start with a hypothesis, you plan an experiment, make a sample, try to collect the data, process and analyze that data, and hopefully the results that you get from the end of that prove or maybe disprove your hypothesis. But one of the things that we thought about in some depth was this experimental planning stage, which is so important. And what we kind of came to the realization of was that you really have to think about your experiment in reverse when you're planning an experiment. So you can start with a hypothesis, but to really make a good design, you really have to think about the results that you will need, sort of the endpoint of your experiment first, in order to what you think the results should be to address your hypothesis. And if you do a good job of nailing that down, then you can start making a good inference as to what is the data that you will need to get those results. And if you can do that, then you can make a good guess as to what are the kind of parameters that you need to capture that data. Do you need to image fast or over large fields of view, et cetera? And only then do you really have a good place to start with selecting your microscope, or in this case, your microscopes. So this I think is important in this context as well, because if we go back to our pyramid of frustration, experiments are going to require maximizing multiple parameters a lot of times to get the full picture. But to choose the optimal combination of microscopy techniques, you really have to kind of resist the urge to just choose the instruments that you happen to have available, or maybe chase after the latest and greatest advanced technologies, but really think critically about what are the instruments that you need to generate that data. And then once you can do that, you can make a decision on how to combine them. Do you just combine results? Do you need to do a correlative, dynamic correlative types of experiments like we just talked about? And I'll just end here by saying that I think it's, especially in this last class of experiments, this is really where the future outlook lies, in my opinion. And that's what I'm calling this event-driven microscopy. And a good example of this was recently published as a preprint, where this group demonstrated this idea where you can actually use biology to tell the microscope what to do. So what they did was labeled some neurons with a ratiometric calcium indicator. And because this was a ratiometric indicator, as they're acquiring this data just in wide field mode on their microscope, they were able to find areas in the sample that seem to be showing some particularly high calcium flux. And this automatically triggers the microscope to then switch modes into in this case, a STED mode, where they can zoom in these areas of interest and start imaging in super resolution mode. And they can repeat this whole process over and over again. So they have this very automated, very biology directed way of doing multi-technique imaging. And I think that that's a very smart way to go about this. And I think it's a very exciting future direction to keep in mind. So I'll end with that. Thank you guys very much for allowing me to chat with you today.
Great. Thanks, Jesse.
Got it.
I don't think I see any questions in the chat yet, but I'll start with one.
Okay.
So you are arguably working at the cutting edge of more microscopy and applications to biology. What do you see as kind of the next big leap in technology forward, or combining of modality?
Yeah, that's a big question. You know, I think microscopy technology kind of tends to progress in fits and starts. I think for a long time, people were very interested in light sheet microscopy and deservedly still are. But I think my last slide kind of points to that question. I think having a microscope that reduces the amount of user input in decision making and really letting the biology decide, do I want to image really fast right now, or do I want to back off and save my photons for later? Those kind of capabilities, I think, is where I think a lot of interesting things going on. And of course, this has a lot of overlap with a ton of work going on in machine learning and coupling machine learning into the actual acquisition, not just the analysis, but the actual acquisition of data on microscopes, I think is a really cool idea.
Very nice, thanks. Any other questions from the audience? No? Okay, I guess with that, I just want to thank everyone for attending and thank again our speakers for 3 fabulous talks. Great work. And hopefully we'll all do this again next year for the next special issue.
Yeah,
Right.
consider submitting next year to our issue.
Yes. Thanks, everyone.